Okay, these are your part 2 of your DNA chapter 8 notes. We're going to talk about chromosome structure. In eukaryotes, DNA is divided into a number of chromosomes that will differ in terms of length and shape. DNA does this and is wrapped around the nucleus only during cell division, usually wrapped in the nucleus unless the cell DNA is usually unwrapped in the nucleus unless the cell is dividing. So how does the DNA wrap up? It will wrap around histone proteins to form nucleosomes, and then those nucleosomes kind of coil around themselves as well. So here's your DNA. Histones are little circular proteins. They will kind of join up and ball up, and the DNA strand will wind around those, and then those histones will kind of stack like beads around each other, but nucleosomes. So each nucleosome is made of a number of histones with DNA wrapped around it, and then the nucleosomes will coil and coil and coil and coil, and that's how your chromosomes are formed. But again, most of the time in the nucleus, the DNA is left unwound that way that it can be copied and replicated and made into proteins and all that stuff, which it can't do if it's all bound up in this way. So how does DNA actually do that replication? DNA replication will occur again during the S phase of interphase. This is an energy-intensive process, so it's something that requires a lot of energy and a lot of focus of the cell, which is why the cell doesn't do much more of life's processes during the S phase. It kind of like pauses everything else so it can focus all its energy on the cell or the DNA duplication. One strand of each molecule is new and the other is parental. So what that means is, in this diagram here, the parental strand is this one on top. It's coming through here. This is the parental strand or the base strand. It tells what the complements will be of the new strand. And then this is also the parental strand. So notice the parental strands come from this bit of DNA that was the old DNA. So you have half of the DNA being old and half of the DNA being new. So if a mutation happened, it's not necessarily going to be displayed the first time around this replicates, because they might still be looking at this old DNA to read and to tell what the genes and the proteins being made are, as opposed to looking at the new DNA. But when it divides again, this new DNA will have its own double strand, and the old DNA again will have its double strand. So 50% of the DNA at the second time would have a mutation that would occur, and then so on. This one and this one would all be mutated with their doubles, and then this one and this one would not be mutated at their doubles. So it would always be 50-50 from there on out. Every cell that came from that would show or exhibit the mutation that would have happened in this case. But let's talk about how this can happen. It occurs in multiple points at the same time. So one chromosome doesn't just decide that it's going to replicate at one end and go to the other end. That would take way too long. The cell wouldn't be able to do this efficiently. So on multiple points on your DNA strand, it's going to perform DNA replication. It is directional. That means there's a leading strand and a lagging strand, as DNA polymerase can only add base pairs to the 3' end. So in my lovely little picture here, let's see if I can get a different color. I want to be blue today. My lovely little picture here, this is my leading strand. It comes through here, and it's leading because it unwinds. So this is the 3' end, this is the 5' end. So it's adding them from the 3' to the 5' end. So this is actually the, oh goodness, I get a little unwrapped in this one. 3' is this one. So it's adding them onto the 3' end of the complementary, or of the parent strand. So the complements are being added in this direction. And this one the lagging strand can only add in the other end. And notice there's a lot more things going on this direction. So it's coming in fragments like this. And those fragments are called Okazaki fragments. So Okazaki fragments on the lagging strand get joined by DNA ligase at the very end of the process. So they have little primers that they get attached to, and they all will be joined together. All these little primer pieces will be cut out, and DNA will be spliced in by a DNA ligase at the very end. So it'll follow along behind this process, kind of zipping it all back together. So the 5 basic steps. The first step is DNA helicase will unwind the DNA. So helicase unwinds the genes. Number 2, Primers will bind with the exposed complementary DNA strands to serve as attachment points of new DNA molecules. So in this one, you would only have one primer that would kind of happen. Oh, here's your helicase, this little unzipper thing right here. It's blue. So in this case, you only have one primer that would bind at the very start where it got unwound, and then it would just read on through. Polymerase would be the guy that comes through and adds the base pairs. But in this one, you have all these primers along the way. So primers will bond with the exposed complementary strands to serve as attachment points for DNA of new DNA molecules. Number 3, DNA polymerase, which is represented by this little yellow box, will move along the DNA strand and add the complementary nucleotides to the new strand in the form of A will bond with T and C will bond with G. Number 4, DNA ligase will follow and seal in gaps so new strands are continuous. So not only is DNA ligase important here in sealing in the gaps on your lagging strand, but it will also follow along on this strand over here and make sure that if you had a section here being transcribed and a section here being transcribed, it'll just, or not transcribed, copied, sorry, replicated, it'll just go through and join all those together as it sees those fragments. So we have a nice smooth single DNA strand. Now I was talking a little bit about mutations. Mutations are mistakes that occur during DNA replication. DNA in a eukaryote will copy 50 nucleotides per second, but in bacteria it copies 1,000 nucleotides per second. So that's a lot of adding in base pairs. I can't even think that quickly. But your DNA will copy that fast. So DNA polymerase is responsible for doing the proofreading and making sure that everything's going well, that there aren't any mistakes, like it'll go back through and follow along again based on what it did. Notice there's 2 different polymerases here, and there's another one that does the proofreading part. So it's specialized to double check to make sure all the bonds are correct. But errors can occur after DNA is damaged as a result of the environment. So things that can cause DNA damage are X-rays, UV rays, gamma rays, different other radiation hitting a cell, or chemicals. And what happens is a lot of these will knock off electrons. They'll knock them around, or they might disrupt the hydrogen bonds that we talked about that hold those base pairs together. So if these hydrogen bonds get broken, and you can't recognize what should go there, you might grab the wrong matching base pair. And so if that happens and it forms a dimer, and I thought I had a picture of dimers here, but I guess I was wrong. But if it forms a dimer, what would happen? Instead of these base pairs being joined to each other across the table, they are now going to be joined to each other like this. They would make hydrogen bonds this way, up and down across the board. And those are called dimers. So dimers will happen every 1 second that you are unprotected in the sun. 50 to 100 dimers will form in your DNA, which is why they say always, 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 always wear sunscreen, because you could be making these dimers now and they might not, in this replication phase or in the next replication phase, they might not have any impact, but like years down the road, as more and more and more of these mutations build up, you might have skin cancer from your choices to not wear sunscreen. So it's not just about not getting a tan, it's about keeping these dimers from forming and keeping your skin cells nice, happy, and healthy. And again, other things can cause these, like other chemicals, like chemicals found in your food, or getting X-rays or gamma rays or any other radiation that you're exposed to can cause your hydrogen bonds to be messed up or electrons to be knocked off, and for mutations in your DNA during replication to happen. So wear your sunscreen, take care of yourself, stay safe, and we'll do new notes next time.